to talk about kind of modelling approach to deriving, let's use the Human Brain Project term, signatures of disease. So what I set out to do a few years ago now was to reconstruct pictures like these pictures on the right here of how a disease plays out over time in terms of the various biomarkers that we use to detect and track those different diseases. So these pictures on the right are hypothetical models of how various biomarkers evolve as the disease plays out. So they come out of the minds of, albeit very well-informed neurologists, to explain which biomarkers show changes first and which track the disease most closely. But they're not directly data-driven in any way. So what I've been set out to do for a while now is to try and learn pictures like this from actual measured data on patients. So these are what I understand by a disease signature, this pattern of biomarker and symptom change over the course of the disease. So you might think, well, it's easy enough to construct a picture like this. All you do is you find a few patients at the start of the disease, and you follow them closely as the disease plays out over time. And then you can make some statistics at each point through that timeline to show how the different biomarkers evolve over time. That doesn't work out for various reasons. The first is it's very difficult to find people at these early stages because often clinical symptoms don't come in until the sort of middle stages of the disease, and a lot of stuff has happened before then that's very informative to as part of the disease signature. A second reason is that it's difficult to make these measurements at very regular intervals. It's at best inconvenient for the patient to have to come in every 2 or 3 months and have a brain scan. But also, some of the measurements that we make are quite invasive, lumbar punctures and so on. You can't really do these things every 2 or 3 months. And a third reason is that the disease can play out at different rates at different patients. So it's hard to place people. Even if you find them all at a common starting point, their symptoms will diverge as time goes on. So in practice, we have to try and reconstruct these pictures from largely cross-sectional data sets. It may be that we have some element of longitudinal data, but largely the challenge is to reconstruct these pictures from cross-sectional data sets. So there what we have is a large number of snapshots sort of at single times through these pictures, but at unknown positions along this progression. And the kind of modelling and learning challenge then is to piece together this larger picture of the disease from this large set of snapshots at unknown points along this time progression. Okay, so that's what I'm going to talk about, various ways of doing that. Why would we want to do this? Well, first of all, I think that reconstructing these patterns of disease gives us some basic biological insight into the disease, gives us a bit of disease understanding. But there are some more pragmatic reasons for doing that as well, in that if we can construct models like this for various diseases, then we can use those models to underpin computer-aided diagnosis and prognosis systems. So just to illustrate that, it's a very sort of simple, idealised example, but let's suppose there are 4 common biomarkers that we measure that are indicative of 2 different diseases. And these are models that we've constructed of these 2 diseases, of how these 4 biomarkers play out or evolve over time. Now let's suppose that a new patient comes in and we measure each of these 4 biomarkers on this patient, and we get these values here, high values for yellow and green biomarkers, and low values for biomarkers blue and red. Which disease do these biomarkers suggest? 1 or 2? Any ideas? Disease 1. Disease 2? Any votes for disease 1? I think it is disease 2. Why is that? Because at no point along this time progression of disease 1 do we have this combination of measurements. High values of green and yellow and low values of of red and blue. There's no position along there that matches, whereas if we compare disease 2, around about here, the sort of middle to late stages, we get a pretty good match, high values of the green and yellow, low values of the red and blue. And we might even imagine having a kind of, a likelihood distribution of this kind of stage in response to measures that we make like that. So not only can these models support a kind of diagnostic system, but they can also help to provide a data-driven staging and stratification system to help identify patients with a particular disease and at a particular stage that you might want to target with some kind of therapy. Okay, so there's the motivation. So let me talk about a couple of traditional approaches to modelling disease progression before I get into the kinds of thing that I want to talk about. So here is a sort of classic old longitudinal imaging study where, so the standard thing to do is then we take a large number of patients at different stages in the disease. We classify them a priori as something like pre-symptomatic, mild AD, moderate AD. So this is a familial AD cohort, so we know which people have the disease but are pre-symptomatic. Then we can evaluate various biomarkers. Here we're evaluating grey matter atrophy at each voxel, compare them against controls, and plot where we see a significant difference. So that's what we're seeing here. And it gives us, really, at the time, this gave us great insight into the way that Alzheimer's disease spreads over the brain, and showed that going from the early stages to the middle stages to the late stages, atrophy spreads over the brain in a very similar way to the way neurofibrillary tangles appear temporally. However, it relies very heavily on this a priori clinical staging, which is done using the MUSE score. Similarly, here's some more recent work, again looking at familial AD. So this is in the DIAN dataset where they measure various biomarkers. And this picture then comes by regressing each of those biomarkers against an expected time to onset of disease. So this is a sort of a kind of an imperfect measure of how far you are through the disease. Basically, it comes from looking at the parent of the patient to see at what age they first presented with clinical symptoms. So first of all, it's an imperfect measure because you don't know exactly when the clinical symptoms started. And then, of course, there can be departures between the parent's time of onset and the child's time of onset. But nevertheless, it does give us some picture of how these different biomarkers evolve. But what we'd really like to do is to avoid having to use any a priori definition of the disease stage. So various reasons for doing that. One is that those disease stage definitions are noisy and subjective. And the second is that they rely on certain biomarkers, which actually we'd like to be able to include in the model itself. But if we're a priori using that for staging, then including it in the model ends up in a kind of a circular argument. Okay, so what I'm going to talk about then is various kinds of models which avoid this circularity and can give us a complete picture of the disease progression over time, or signature. So I'm going to focus on the event-based model, part of a broader class of discrete models, because that's the thing we've been working on most at UCL. But then I'll also mention a couple of other kinds of model which also tick those boxes, so fall into the same kind of category. And these are all things which have really evolved over the last 2 or 3 years with the availability of data sets which are large enough to support them. But most of what I will say is summarised actually in this article here, and Alex, of course, is here, so you can grill no more. Okay, so the event-based model. So this models the disease as a series of discrete events, which typically are biomarkers switching from a normal value to a detectably abnormal value. And then basically what it does is to try to estimate the order in which those events occur from a cross-sectional or short-term longitudinal data set. So just to explain what I mean by short-term longitudinal, by that I just mean longitudinal enough that we can estimate things like grey matter atrophy, so we've got at least 2 close-ish time points, but not longitudinal enough that we can actually map out the whole trajectory of a biomarker through the course of the disease. So basically these are cross-sectional data sets, but they might also have atrophy, but they're cross-sectional snapshots of the atrophy. How can this possibly work? Well, let me try and give you a bit of intuition as to how you can estimate this longitudinal information from cross-sectional data. Let's think just about two biomarkers, a very simple problem. So here they are, red biomarker and blue biomarker. And we can see that at the start of the disease on the left there, both have a normal value which is around 1, and at some point both switch, but we can see that the red one goes before the blue one. Now let's take a bunch of patients, here they are. So the values on the top are values of biomarker, the red biomarker. Slightly confusingly, in this picture, red means a high value, blue means a low value. But this is the red biomarker, this is the blue one. So let's look at a few of these patients. So here's one who has a high value of both biomarkers. That doesn't tell us an awful lot about the ordering, it just tells us that they're somewhere near the beginning of this progression. Here's another one who has a low value of both biomarkers. Also doesn't tell us much about the ordering, just that they're near the end. But it's people in this middle zone who have a high value of one biomarker and a low value of the other that inform about the ordering of these biomarker changes. The more people we have that have a high value of blue biomarker and a low value of red one, the more evidence we have that the red one goes before the blue one. And that idea scales up to a larger number of biomarkers, and that's basically the way the event-based model works. So we look for people who have got normal values of 1 but abnormal values of others. Yes?
Is life so nice that biomarkers always monotonically increase or decrease?
So yeah, so there is an assumption in here that these are one-way streets, that things are always degenerative and that there's no remission. Yes, life is not so nice, but that's the way my model is that. Slightly scary slide. Sorry about this one, but just to give you an intuition to the mathematics of this. So the way the model works is then that we formulate a way of evaluating the likelihood of a particular sequence of events so that we can then find the sequence that maximizes that likelihood. Just talk you through this briefly. So we can formulate an expression for, so the top line is the probability of biomarker value, the value of the ith biomarker in patient J, given that we know the sequence of events and that this person is at stage K of this sequence of events. Being at stage K means the first K biomarkers of the sequence have gone abnormal, the next N minus K are still at normal values. Okay, so we're going to assume then that we've got models for the distribution of values in the normal and abnormal states. Given those, then we can estimate this probability of a single data point given the sequence and the stage. We can integrate out this hidden stage variable to give us a probability of the data point given the sequence, and then just multiply over all the different patients contributing data to give us the probability of a whole data set from a load of patients given a particular sequence of events. Then all we try and do is look for the sequence that maximizes this probability, and that's our estimated, our best guess sequence. But we can also evaluate the likelihood of any sequence to see how much we believe the most likely one. Let me jump straight to the kind of output that this gives us. So these were the first results we got from using this model. And the set of events were the appearance of atrophy in lots of different cortical brain regions. So what the output then is a list of brain regions in which atrophy occurs, ordering them by the time that atrophy occurs. And so we can see this mapped back onto the brain. This is a map for Alzheimer's, this is one for Huntington's. So there's some nice results come out of this in that we see the kind of memory-related areas going early in Alzheimer's, but being spared quite late in Huntington's. And on the other hand, motor areas are spared quite late in Alzheimer's, but go early in Huntington's, which is what you'd expect for these two different diseases. The other nice thing that you get is this we call it a positional variance diagram, but it's essentially a posterior distribution on the event ordering. And what this, so it's an uncertainty on that, on the best guess event sequence. And what this tells us is where we believe strongly in the ordering, so the first 3 events here are very strongly ordered, but then the next 5 or so are actually pretty much interchangeable. But they all come after these first 3, and they all come before this big block of 20 or so down here. So it helps us to interpret where we believe strongly in the ordering that we get back and where we're not so sure of it. So Alex then picked up this work later, and it took a while actually to reformulate this model for application to the ADNI dataset. The difficulty there is that we don't have a well-defined control group, and that makes the modelling the underlying biomarker distributions a bit harder. Eventually we overcame that and used it in a slightly different way to compare a wider variety of different biomarkers, CSF biomarkers, imaging volumes and atrophies, and various cognitive tests. And so this is the positional variance diagram that came out of that.
Sorry, I didn't realize why is it the absence of a control so critical for you?
Yeah, so I glanced over something narrowly back here, which is how we estimate the probability of a biomarker value given that the event that has occurred and given that it hasn't occurred. So it's very easy to estimate probability of value given the event hasn't occurred if you've got a well-defined control group, because they can just get it straight from there. If you haven't, it's much more difficult to estimate that probability. And so you have to use a kind of more robust statistical techniques and mixture modelling to try and pull out those 2 distributions. So that's essentially the difference in the model for a sporadic disease. Okay, so that model also provides then a staging system. We can classify new patients at any of the stages defined by that ordering. And a nice validation of that is that so we get 15 different stages because there are 14 biomarkers. And so a nice validation that this model is doing something sensible is that it pushes all the AD subjects to the late stages and all the cognitively normal, so most of them at least, to the early stages. And similarly, gives you a slightly weaker but nevertheless clear separation between MCI converters and stages. And a Cox proportional hazards analysis shows that staging is a strong predictor of conversion of MCIs to AD or cognitively normals to MCI. So the very latest on this, it extends the model to capture heterogeneity in the progression sequence. So the original model assumed that everybody underwent the same progression. Fairly clearly not true. What the extended model that has just been accepted to the ITMI conference this year. So this will try and pull out subtypes from the data set which undergo different event sequences. So that's a very nice development, I think, that scales up very naturally to address some of the questions that the earlier speakers outlined of trying to pull out data-driven disease signatures and not being tied to the traditional labels and diagnoses. Okay, so the other models I just wanted to mention, kind of limitation of the event-based model is it's very discrete. It just says things initially normal, suddenly they switch to abnormal. Obviously that's not really the way biomarkers behave. There are various other models which can learn a more continuous trajectory of biomarkers, again from largely cross-sectional data or short-term longitudinal data. Differential equation models use short-time longitudinal data points to estimate and give lots of, across a large number of patients, to give lots of samples of the gradient of a biomarker trajectory. And from that you can piece together a long-term picture of how it evolves. Stochastic differential equation models, very similar, but allow you to then model the dispersion in those trajectories. The limitation of these is that they treat each biomarker independently, and so they don't, pull out dependencies among different biomarkers. But other recent work on self-modeling regression will try and find a complete picture of the way all the biomarkers evolve with respect to one another simultaneously in order to pull out a more complete picture. So this is the latest work from Mike Donoghue showing this kind of thing at the AAIC conference last year. Final class of model I wanted to talk about is spatio-temporal models. So they've a slightly different aim here, which is to pull out a picture, a 4D picture of how a disease affects the brain. So the aim would be to construct almost a movie of the sort of average trajectory of the brain over time undergoing some process, normal aging or a particular disease. So it's a bit like the earlier work I showed you with classifying people a priori into mild, moderate and severe classes and just seeing how they differ. But the latest ways of doing this can avoid that a priori classification. So you have to account for the fact that people are going through the disease at different rates, but there are various ways of getting around that problem and trying to find the trajectory that best fits a whole cross-sectional data set and simultaneously estimating the position of each of your cross-sectional subjects. So it's a tricky problem, but with a large enough data set you can do it. Okay, so that's all I was going to talk about. So I'll just give you some pointers towards our ongoing and future work. So I mentioned this issue of progression heterogeneity. All of the models I've talked about basically assume that there's a single trajectory underlying all of the data. And I think getting away from that simple assumption is a big thing to look at in the future. As I've shown you, we've made some first steps towards doing that. Another thing that these ideas lend themselves quite nicely to is getting around the problem of missing data. So, in our first forays into this idea, we'd have to limit the amount of data we could use to inform the models to ones where every patient had all of the biomarkers that we wanted to model. But actually the formulation of the model is reasonably robust to some of that data being missing. And sort of recently, Alex again has been exploring the effects of this. And actually I think it turns out that the models are quite robust to having missing data if you treat it the right way. And that's very nice because it means we can massively increase the number of data points that we use to inform the models, which is not an option for some other ways of analysing these data sets. Another big assumption that the models make is independence of the different biomarkers. This is not always true. So even at a fixed time point in the disease progression, so for example, the volumes of the hippocampus and the neighbouring entorhinal cortex are very likely to be highly correlated. Our model assumes they're not. And so this again is something that we've been studying the effects of, and I think the conclusion is that it doesn't have a huge effect on the sequence that we estimate, but it does affect the confidence in that sequence. We tend to overestimate the confidence. Questions there about how to proceed. Do we try and model the correlations, or do we just learn what the effect is and deal with it? The problem with the former is that it massively increases the number of parameters we have to estimate. Including static variables, so things like encoding the dependence on genetics and demographics and things is another challenge at the moment. We've done that only in very simplistic ways. By differential models, I mean learning, factoring out things like the normal aging process, which we've made no attempt to do so far. And longer term, I think what we're looking to do is to apply these ideas to a wider set of neurodegenerative diseases so that we can actually start to do what I talked about at the start and build computer-assisted diagnosis systems built on a range of models for different kinds of diseases.